Hello, my name's Zachary, and today I'll be talking about the potential of a specific enzyme in changing bacterial composition in a mouse model of colitis. Firstly, the human body hosts trillions of microorganisms, their genes, and synthesized products, collectively called the microbiome. This microbiome plays a critical role in maintaining host health, and its optimal functioning depends on rich species diversity as well as balance. Unfortunately, when this balance is disrupted, also called dysbiosis, it can lead to various diseases, including colitis, which in 2023 was estimated to have 5 million cases worldwide. Knowing the importance of the microbiome to many different disease states, we have chosen to look into bacteria, one of the most prevalent within the microbiome. It turns out that bacteria are able to communicate using small molecules called autoinducers. In this depiction, triangle represents an autoinducer, which is produced inside the cell and then diffuses outside. These specific autoinducers are called acyl homoserine lactones, and when they accumulate in great enough density, they diffuse back into the cell, bind a receptor, which binds the promoter and results in gene expression. This gene expression may elicit behaviors ranging from creation of biofilm to release of virulence factors, which can be harmful to the host. So what this project focuses on is disrupting bacterial communication and seeing what it does to the disease. These blue square-looking things are quorum-quenching enzymes, which we use in this experiment and are used to degrade these AHLs. When AHLs are degraded, they cannot enter the cell, bind the receptor, bind the promoter, and ultimately gene expression is canceled. So how did we assess this effect of quorum-quenching enzymes in a colitis model? Well, we had 20 male mice and 20 female mice treated with either regular unamended drinking water or SsoPox, which is one of these quorum-quenching enzymes that I mentioned. We let these mice drink these treatments through sipper bottles for 3 weeks while collecting feces weekly. At experimental week 3, dextrans sodium sulfite, or DSS, was added to these sippers, and which is commonly known to induce colitis in mice. At experimental week 5.5, the mice were euthanized, and the feces that we had collected amongst or throughout the entire study was used to extract DNA, which underwent 16S RNA sequencing and then data processing to create figures and analyze the results. So we'll start off looking at relative abundances in females. On the y-axis here, we have mean relative abundance. Basically, a bigger section of color indicates there was a greater abundance of a certain bacteria within the sample. So what we're seeing here is at week 0, between the drinking water and SsoPox mice, as expected, there's not a significant difference. Now moving on to week 3, once these mice have been treated with the SsoPox for a significant period of time, we now see that Turkibacter is significantly decreased in SsoPox mice, suggesting a potential effect. Now, after inducing DSS, we see that there are no significant differences between the drinking water and SsoPox mice, meaning that DSS might have more power or it overcomes the effect of these quorum-quenching enzymes. A similar story holds true in males, where at week 0, there are no significant differences in composition, or relative abundance rather. But then at week 3, we see a different bacteria, significantly different abundance between the treatment groups, this time Bifidobacterium. But then the same story holds true where once we get to week 5, where the DSS is going to work, there are no significant differences between the drinking water and SsoPox. We can also look at a principal coordinate analysis, which is a matrix of dissimilarity. Essentially, each dot represents one mouse and its microbial community. Dots that are closer to one another are more similar in community. And we found in females that there were not significant differences between beta diversity, or we can say this is a proxy for community composition between the drinking water and SsoPox groups at the same time point, meaning that the lighter ones are near the lighter ones and the darker ones are near the darker ones. A similar story was seen in males, although at week 3, there was significant separation where we're seeing these pink ones in the middle are clustering toward the right, whereas these lighter blue ones are clustering toward the left, suggesting a significant difference in community composition between the drinking water and SsoPox at week 3 only. So what we found is that SsoPox appears to change microbial composition in mice. This is evidenced by those first 3 weeks where we're seeing some changes. And also there's sex-dependent effects. If there weren't, we would expect to see more or less the same results for the males and females. Now, moving on to later in the experiment, we see that there are no significant differences in the relative abundance of taxa or community composition at week 5 between the groups, which suggests that DSS significantly changes microbial communities regardless of quorum quenching treatment. As for some conclusions, we can say that the 1 milligram per milliliter treatment of SsoPox does not significantly alter microbial composition after colitis is induced by DSS. However, SsoPox does show a promise in its ability to shape microbial communities, as evidenced by the week 1 to 3 results. For future directions, I'm wondering if a higher concentration of SsoPox, for instance, 2 or more milligrams per milliliter, will change these results. Maybe it can override DSS. There's my references. Thank you. If you have any questions, feel free to email.